Okay, so a very warm welcome to all esteemed guests, distinguished speaker, and fellow speakers. I'm delighted to welcome you all to this one-day international webinar on molecular genetic approaches in wildlife conservation, organized by the Department of Conservation Biology in collaboration with Internal Quality Assurance Cell, Durgapur Government College. This webinar brings together a platform to discuss the advancements in molecular genetics and their pivotal role in safeguarding the planet's precious wildlife. The topic also extends its potential in addressing the critical challenges faced in wildlife. Since Dr. Devnath Palit, patron of this webinar and principal Durgapur Government College, is unable to attend due to some unavoidable engagement, he has sent his best wishes for the success of this webinar. Without further ado, I would now request Dr. Tapujit Bhattacharya, Assistant Professor, Department of Conservation Biology, Durgapur Government College, to kindly take the privilege to introduce our honorable speaker with us.
Thank
Over
you,
to you,
Sanchari.
sir.
It is my pleasure to introduce Dr. Supriyo Dolui, an exceptional conservation geneticist dedicated to protecting the Himalayan wildlife in India. Dr. Supriyo's research journey began as a project intern at the Wildlife Institute of India in 2016, where he joined our project with immense enthusiasm and dedication. Since then, he has grown into a prominent figure in conservation genetics, focusing on landscape genetics, habitat modeling, and conservation genomics. Dr. Supriyo completed his PhD at the Geological Survey of India, investigating the impact of past geological events on the demography, distribution, and genetic makeup of the red panda in the Indian Himalayan region. He holds an MSc in Zoology with a specialization in wildlife and conservation biology from the University of Calcutta and a BSc in Zoology from Scottish Church College. His academic excellence is further demonstrated by his performance in national examinations, securing top ranks in CSIR UGC NET and GATE Ecology. Dr. Supriyo has acquired a diverse set of skills throughout his career, including molecular biology techniques like isolation of nucleic acids, PCR, and genotyping, as well as computational expertise in remote sensing, GIS, climate change modeling, and population genetics software. His proficiency in both wet lab and computational methods makes him a versatile researcher. His dedication to uniting science and local communities to preserve biodiversity is truly inspiring. Dr. Supriyo's passion for advancing research on the fauna of the Indian Himalayas and his commitment to cutting-edge genomics tools and techniques have been recognized through numerous awards and scholarships. Notably, he received the International Travel Grant from the Science and Engineering Research Board (ACRB) and the individual project grant from the Mohammed bin Zayed Species Conservation Fund. Currently, as a postdoctoral fellow at ETH Zurich and the Swiss Federal Research Institute (WSL), he continues to push the boundaries of genomics research. With numerous publications and contributions to books and conferences, Dr. Supriyo has established himself as a leading expert in this field. Please join me in welcoming Dr. Supriyo Dolui. Over to you, sir.
Um, Sir, before our honorable speaker begins, I would request all the participants to kindly be muted throughout the presentation. And if you have any question, you can drop it in the chat box, or at the end of the presentation, we'll open the session for discussion. Over to you, sir. Thank you.
Thank you, Dr. Tapajit Bhattacharya, sir, for introducing me so well. And I feel happy that after so many times I get to hear you. Yes, last time we met in WII in 2016, and you remember that. And hope you are doing good, sir. And I would like to thank, first of all, my friend and colleague, Nasma Khatun, for inviting me for this talk. And but still, I am grateful to the principal of this college, Dr. Devnath Palit sir, and head of the department, Dr. Maitri Chakraborty ma'am, for giving me the invitation for presenting my talk to the students of your department. I'm really glad to have this opportunity. So, as Dr. Tapajit sir mentioned, I just finished my PhD last year from Zoological Survey of India and with Calcutta University. I did my PhD work on red panda, and for that I spent like 6 years in the eastern Himalayan part of India, and it was a great journey. Conservation biology is something which inspires you not just about the wildlife, it's about the experiences you gathered all together during your PhD, the travels to the different parts of India, meeting new people there and sharing their experiences. It all together broaden your mind. So I'm lucky that I got this opportunity to do my PhD in the Eastern Himalayas, and as Tapaji sir mentioned, right now I'm doing my postdoc research that is on alpine ibex conservation in Switzerland. So I'm currently associated with the ETH Zurich and WSL, the institute, and this is under a scholarship program which is Swiss Government Excellence Scholarship. So this is for a 1 year postdoc. So right now I'm working on the alpine ibex. So I believe I am audible and visible properly to all of you, right?
Yes, sir.
Okay. So without ado, should I start my presentation?
Yes, sir.
Thank you. And I would like to present in both Bengali and English so that it's like it can be like a conversation or just like a storytelling, because I believe all of the students are from MSc background, so it might not be possible for them to understand all the complex genomics tools and techniques. So I try to keep it simple so that they can understand what are the genetic approaches we are working right now, what are the future in this field of molecular genetics in wildlife. So let me just share my screen. Okay, can you see my screen? Can you see my presentation?
Yes, sir. It's visible.
Okay, thank you. So today I'm going to present my topic called molecular genetic approaches in wildlife conservation. So first of all, what is conservation genetics? So, conservation genetics is particularly a multidisciplinary field of study যেখানে আমরা জেনেটিক ইনফরমেশন অ্যাবাউট ওয়াইল্ড লাইফ পপুলেশন এবং ওয়াইল্ড লাইফ জেনেটিক্স এবং ইকোলজি সবগুলোকে একসাথে কম্বাইন করে এটা বোঝার চেষ্টা করি যে হোয়াট ইজ হাউ টু প্রিজার্ভ দি বায়োলজিক্যাল ডাইভার্সিটি ইন আওয়ার ওয়ার্ল্ড অর ইন আওয়ার সারাউন্ডিং নেচার. So there is an interconnection with population genetics. So the main aspect of conservation genetics actually relies on the principles of Hardy-Weinberg equilibrium theory. So যেটাকে আমরা population genetics বলে জানা থাকি, তো population genetics এর basic principle যেটা Hardy-Weinberg propose করেছিল সেটার উপর বেস করেই আমরা কনজারভেশন জেনেটিক্স এর কাজ করে থাকি, যেখানে আমরা চেষ্টা করি এটা জানার যে একটা ওয়াইল্ড পপুলেশনের জেনেটিক ডাইভার্সিটি কিরকম, is there any kind of threats that the population are facing right now or not. In the concept of phase, I am saying like, is there kind of inbreeding depression? Are this population vulnerable for any kind of wildlife diseases? And based on these principles of Hardy-Weinberg theory, we try to develop some strategies to preserve this wild population so that they can sustain in the nature for a longer period of time. So the key principles and objectives of conservation genetics are to understand the genetic factors affecting population health, and to develop strategies to preserve and restore genetic diversity. So to understand the genetic factors affecting population health means what are the genetic factors? I mean, that can be like genetic diversity, if the population is suffering from like homozygosity or from inbreeding depression, is there any kind of vulnerability towards wildlife diseases in the nature? So this kind of genetic factors which can affect the population health in the nature, we can assess this by the help of conservation genetics. And then developing strategies to preserve and restore genetic diversity. It means like first we have to understand the population's genetic diversity or the genetic status of that population, and then we can plan for different strategies to protect these populations in the wild. And how we can do that? We can try to find out the gene flow pathways, how the populations are connected in the wild, and then we can try to protect this particular connectivity or the corridors that this wildlife uses so that their genetic diversity can be maintained. So this is how we can use the principles of population genetics to conserve the wildlife species. So now I would like to discuss in detail about the Hardy-Weinberg equilibrium. So basically, these are two scientists, Hardy and Dr. Weinberg. They're not at all biologists or ecologists in their in their work. Dr. Hardy was an English mathematician, and Dr. Weinberg, he was a German physician. And both of them, they proposed this principle for wildlife and population for the wildlife. So their principle says that Hardy-Weinberg equilibrium is a fundamental concept that describes the idealized genetic state of a population where allele frequencies remain constant from generation to generation. So basically, they proposed a theory. This is a principle, this is a hypothesis. It may not be, it will not reflect in the actual natural world. So যেরকমটা mathematics কোনো hypothesis হয়ে থাকে, so they imagined an idealized population. The population should be conditions for equilibrium are maintained under specific conditions. Those are like large population size, no mutation, no migration. There should be like random mating and no natural selection. So let me just draw a simple diagram so that students can understand. So what they imagined a large population. So this should be a closed population, a large population, and there should be a large number of individuals in this population. So imagine this circle as a huge population, and these are the individuals in this population, under this population. So according to their theory, the genetic variation or the allelic frequency of these individuals in this population will remain same if no external forces are acting on this population. What kind of external forces they are mentioning? Like there should be like no mutation. So all the individuals, they will not go through any kind of mutation. Then there would be no migration. So no outward migration from this population and no inward migration towards this population. Because if there's migration going to happen, then the population demography will change. Then there will be like random mating. So these individuals can mate with each other randomly. There will be no mate selection process. But we all know that in nature, the species, they select mate based on specific traits. And the last was that no natural selection. Natural selection is an ongoing process. It's an evolutionary process. It always happens. But they imagined a hypothetical population in which these all external forces doesn't work. So based on this, they have proposed this theory of Hardy-Weinberg equilibrium. And what we actually want to study in population genetics or in wildlife genetics is that is a wild population, how much it is deviating from the Hardy-Weinberg equilibrium. So the formula or equation for Hardy-Weinberg is like p square plus q square plus 2pq equals to 1. So the genetic frequency or the gene frequency will remain 1. But in actual scenario, in actual world, this frequency changes from this equilibrium. So it is our job to understand how much of this frequency is changing in the nature, and because of this, is the population going towards any threat or not. So yes, as I have mentioned, these are the top 5 conditions they have mentioned for an idealized population, which is maintaining a constant genetic variation. No selection, no mutation, no migration, large population, and random mating. These 5 are important points. So before going into the details of Hardy-Weinberg theory, so just a basic recap of what is gene, allele, genotype, and phenotype. So genes are segments of DNA responsible for specific traits. So if you consider these as a segment of DNA, you can see there is a particular gene in this location. For we have a pair of DNA. So in both pair of DNA, there will be one set of these alleles. So what are alleles? Alleles are different versions of a gene. So they are so this is, if this is a DNA fragment and this is a gene, so the alleles can be the different versions of this gene. ছোটবেলায় আমরা মেন্ডেলিয়ান থিওরি পড়েছিলাম, যেখানে আমরা পড়তাম যে ক্যাপিটাল টি স্মল টি, তো দিজ আর লাইক দি অ্যালিলস। সো ক্যাপিটাল টি অ্যান্ড স্মল টি সেক্ষেত্রে হচ্ছে alleles. So for example here, gene A with 2 alleles, that is capital A and small a. So, and these alleles can have different combination for them, like capital A hote pare, capital A small a hote pare, small a hote pare. So this is a dominant allele, this is a kind of heterozygous allele, and this is a combination of recessive alleles. So এই জিনোটাইপগুলো যখন কোনো একটা ফেনোটাইপকে প্রকাশ করে, তখন আমরা সেই পার্টিকুলার জিনোটাইপটাকে সেই ফেনোটাইপের সাথে অ্যাসোসিয়েট করি। তো যদি একটা কোনো ডমিনেন্ট অ্যালিল হয়ে থাকে দুটো, সেক্ষেত্রে যেমন যদি হয় যে হাইটের ক্ষেত্রে আমরা দেখছি সবগুলো ইন্ডিভিজুয়াল লম্বা হচ্ছে। হেটারোজাইগাস যদি হয়ে থাকে একটা ক্যাপিটালে একটা মানে একটা ডমিনেন্ট অ্যালিল এবং একটা রিসেসিভ অ্যালিল, সেক্ষেত্রে যে আউটকামটা প্রকাশ পাবে ফিনোটাইপের, সেটা হয়তো লম্বা হবে, বাট ইট উইল হ্যাভ আ রিসেসিভ অ্যালিল ইন দেয়ার জিন। আর যদি রিসেসিভ হয়ে থাকে সেক্ষেত্রে সবগুলোই তাহলে বেটে হচ্ছে। সো বেসিক মেন্ডেলিয়ান কনসেপ্ট। তো আশা করি সবাই বুঝতে পেরেছে যে হোয়াট ইজ জিন, হোয়াট ইজ অ্যালিল, অ্যান্ড হোয়াট ইজ genotype and phenotype. So role in genetic variation and inheritance. So genes and alleles determine genotype, which in turn influences the phenotype and contribute to the genetic diversity within a population. So this is very important to understand because, as I have mentioned, for a wild population to survive in the nature, it is very important that they should maintain a heterozygous conditions. যত বেশি homozygosity হবে, মানে same kind of alleles হবে, তত বেশি they will be vulnerable to different kind of anthropogenic threats or natural threats. But যদি population mainly heterozygosity maintain করতে পারে in wild, then they can survive in the nature for a longer period of time. So now let's dive into the detail of the Hardy-Weinberg equilibrium. So these are the 5 key concepts of Hardy-Weinberg, and I would like to highlight all of these concepts briefly. So first of all, migration. So there are like two kinds of migration, that is immigration that introduces new genetic diversity. So যখন কোনো পপুলেশনের বাইরে থেকে কিছু ইন্ডিভিজুয়াল আসছে, সেটাকে আমরা বলছি immigration, and in this case সেই বাইরের ইন্ডিভিজুয়াল গুলো they are introducing some new kind of genetic variation to this already existing population. তাহলে সেক্ষেত্রে কি হচ্ছে, introduces new genetic diversity into a population and genetic diversity সেক্ষেত্রে increase হচ্ছে. And then what is immigration? Immigration reduces genetic diversity in a source population. তো একটা সোর্স পপুলেশন থেকে যখন কিছু ইন্ডিভিজুয়াল বাইরে মাইগ্রেট করে চলে যাচ্ছে, they are taking some particular genetic traits or genetic variations with them from outside, from the population to outside. তার ফলে কি হচ্ছে যে এই যে বেসিক পপুলেশনটা ছিল সোর্স পপুলেশন, তার জেনেটিক ডাইভার্সিটি কমছে। So the balance between immigration and immigration actually influences the genetic diversity within a population. তাহলে হার্ডি-ওয়েইনবার্গ থিওরিতে যে প্রথম কনসেপ্টটা ছিল যে কোনো মাইগ্রেশন হবে না, বাট ইন রিয়েল ওয়ার্ল্ড উই সি লাইক দেয়ার আর ডিফারেন্ট পপুলেশন অ্যান্ড দেয়ার আর লাইক immigration and immigration between these populations happening. So the genetic diversity is not constant. The genetic variation is always changing because of these external forces like migration. One of the fine concept of migration is founder effect, যেটা আমার নিজের খুবই personally প্রিয়. The founder effect occurs when a small group of individuals establishes a new population. So population সবসময় চেষ্টা করে, if there is a source population or a big population, so the individuals of this big population always try to migrate out from this source population to the outside population or to a new area where for the search of better resources. তো যখনই একটা এরিয়াতে অনেক বেশি ইন্ডিভিজুয়াল হয়ে যায়, তখন কিছু ইন্ডিভিজুয়াল চেষ্টা করে যে বেটার রিসোর্সেস এর জন্য বাইরে নতুন কোনো জায়গায় গিয়ে কলোনি এস্টাবলিশ করার। তো সেটা যখন হয়, there would be only few individuals from the source population who will travel out to a new area or to a new location for establishing a population. তো in that case কি হয় যে কিছু কিছু জেনেটিক ভ্যারিয়েশন লস হয়। If we see at this diagram, তাহলে দেখা যাবে যে এখানে 3 ধরনের ইন্ডিভিজুয়ালস ছিল, the red, black and white, but only 2 types they migrated out to this new area, the black and the red variation. তাহলে এই ক্ষেত্রে কি হলো যে কিছু জেনেটিক ভ্যারিয়েশন লস হলো, because a particular white variant they didn't migrate out to this new population. তাহলে এইখানে যে নতুন কলোনিটা তৈরি হবে, they will not have this white butterfly, so there will be a less genetic diversity which may have been introduced by this white butterfly to this new colony. So founder population often have reduced genetic diversity, as it is mentioned, and increased vulnerability to environmental changes. যেহেতু ডাইভার্সিটি কমে যায়, সেক্ষেত্রে যে কোনো ন্যাচারাল ক্যালামিটিস হোক বা যে কোনো অ্যানথ্রোপোজেনিক প্রেশার হোক, it can hamper the new population. Then there's this concept of mutation. Hardy-Weinberg equilibrium theory second concept is mutation. So mutation is a fundamental source of genetic variation. Mutations are changes in DNA sequences that can result in new alleles. So mutation is something that is very constant. Hardy-Weinberg equilibrium যেটা বলা হয়েছে যে মিউটেশন যেন না হয় একটা পপুলেশনের মধ্যে, but mutation is something which is always happening in all the animal world. So mutation কে থামানো অসম্ভব. So পপুলেশন জেনেটিক্সের যে মেইন মিউটেশনটার কথা আমরা ডিসকাস করে থাকি সেটা হচ্ছে সিঙ্গেল নিউক্লিওটাইড পলিমরফিজম। সো দেয়ার আর উই নো যে 4 কাইন্ড অফ নিউক্লিওটাইডস ATGC। সো এবং তারা পেয়ারে অবস্থান করে, সো এ এন্ড তার সাথে হচ্ছে টি। সো সিঙ্গেল নিউক্লিওটাইড পলিমরফিজম ইজ যখন একটা নিউক্লিওটাইড মিউটেশনের জন্য চেঞ্জ হয়ে যাচ্ছে। সো ইফ ইট ওয়াজ লাইক A and T, সো সিঙ্গেল মিউটেশন সিঙ্গেল নিউক্লিওটাইড পলিমরফিজম হলে এখানে হয়ে যেতে পারে A to C। সো এই যে একটা সিঙ্গেল চেঞ্জেস এলো, তার ফলে কি হলো, একটা ইন্ডিভিজুয়াল বিকাম সেপারেটেড ফ্রম দি আনাদার ইন্ডিভিজুয়াল। সো তার মধ্যে নতুন কিছু ভ্যারিয়েশন সৃষ্টি হলো। এরপর যদি একটা পপুলেশনের মধ্যে এরকম হয়ে থাকে যে দুটো পপুলেশন রয়েছে এবং তার মধ্যে একটা পপুলেশনে কনস্ট্যান্টলি মিউটেশন হয়েছে এই A to C, এবং একটা পপুলেশনের মধ্যে সেই যে প্রিমিটিভ বা সোর্স নিউক্লিওটাইডটা ছিল সেটাই থেকে গেছে, দ্যাট ইজ A and T। সো তার ফলে কি হবে যে ওভার দ্য পিরিয়ড অফ টাইম this and this population will become separated from each other and they may give rise to different phylogenetic species, যেটা এখন আমাদের ওয়াইল্ড লাইফ কনজারভেশন একটা খুব হট টপিক, that is the phylogenetic species concept. I'll discuss about this later in my slides. So that basically happens because of accumulation of this kind of single nucleotide changes. So obviously Heisenberg theory অনুযায়ী mutation হওয়ার কথা নয়, but nature always mutation is happening and it is introducing some kind of genetic variation to the populations. Then another interesting phenomena of the nature is bottleneck theory. So bottleneck theory is a very interesting concept. So that represents this kind of bottleneck situation. So what is this? So bottleneck occurs when a population undergoes a drastic reduction in size. Genetic diversity is lost as a result and the population becomes genetically homogeneous. Bottlenecks can be caused by natural disasters, habitat fragmentation, or human activities. Understanding and mitigating bottleneck is crucial for preserving genetic diversity. So if we imagine a single parent population which have a homogeneous number of individuals, like here you can see like blue and yellow dots, they are like in equal numbers. So if suddenly a drastic calamity happens, some kind of natural disaster happens, so what happens যে কিছু ইন্ডিভিজুয়াল এখান থেকে সার্ভাইভ করতে পারবে এবং মোস্ট অফ দ্য ইন্ডিভিজুয়ালস দে উইল নট সার্ভাইভ দিস ন্যাচারাল ক্যালামিটি। তো তার ফলে যে নতুন ফাউন্ডার পপুলেশনটা তৈরি হবে, দে উইল হ্যাভ লাইক ডিফারেন্ট ডেমোগ্রাফি from their parent population. Like in this scenario, if we see, there are like more number of blue balls than the yellow balls, and they will be the surviving individuals who will give birth to the next generation. And in the next generation, you can see that the population demography has changed. Here, more number of blue individuals than the yellow individuals. So this is how bottleneck occurs in nature. নেচারে কোনো একটা অ্যানিমেল পপুলেশন হয়তো হিউম্যান ফ্র্যাগমেন্টেশনের জন্য পকেট পপুলেশনে পরিণত হয় এবং সেখানে হয়তো কিছু ইন্ডিভিজুয়াল সার্ভাইভ করে এবং সেখান থেকে নতুন একটা পপুলেশন তৈরি হয় এবং দে হ্যাভ রিডিউস জেনেটিক ডাইভার্সিটি বিকজ অল অফ দ্য ইন্ডিভিজুয়ালস দে ডিডন্ট সার্ভাইভ দি প্রিভিয়াস ক্যালামিটি। সো বটলনেক এবং ফাউন্ডার এফেক্টের জন্য যেটা হয়ে থাকে, দ্যাট ইজ কল্ড জেনেটিক ড্রিফট. So genetic drift is the random fluctuation of allele frequencies in small population. So if there is a very small population in the nature and সেখানে যদি আমাদের ফাউন্ডার এফেক্ট বা বটলনেক হয়ে থাকে, যার ফলে কি হচ্ছে যে কিছু জেনেটিক ভ্যারিয়েশন লস হচ্ছে বিকজ অফ ফাউন্ডার এফেক্ট, কারণ তারা একটা পপুলেশন থেকে বেরিয়ে অন্য জায়গায় চলে যাচ্ছে, বা বটলনেক এফেক্ট, যার ফলে কি হচ্ছে যে কিছু ইন্ডিভিজুয়াল আর নট সার্ভাইভিং বাট অনলি ফিউ ইন্ডিভিজুয়ালস আর সার্ভাইভিং and giving rise to the next generation. So that incidence is called genetic drift, and genetic drift can lead to loss of rare alleles and reduce genetic diversity. This is a very important point because rare alleles are actually important for survival of any kind of population, because these are the rare alleles who contribute some different kind of variation to the genetic component of an individual, and that is why we may survive different kind of diseases or different kind of natural changes. So in those scenarios, these rare alleles become activated and they help to combat those particular natural disasters and diseases. So if there is a loss of rare alleles, so it becomes difficult for the population to survive in the changing environment. Then the last concept is the natural selection. Heisenberg's concept অনুযায়ী, principle অনুযায়ী, there should not be any kind of natural selection to the large population they have hypothesized. But in nature, we know that natural selection is a fundamental driver of evolution. Evolution is an ongoing process, and evolution হওয়ার জন্য natural selection সবসময় হচ্ছে এনভায়রনমেন্টের মধ্যে. So there are three kind of natural selection in process. So the first one is stabilizing selection. So what is stabilizing selection? Stabilizing selection favors the intermediate phenotypes, maintaining genetic diversity within a population. So if we look at the first graph, so if this blue line is a population, only the intermediate phenotype or the intermediate variations will survive for natural selection. So এই ক্ষেত্রে যেটা example দেওয়া আছে যে রবিন পাখির ডিম. So robins typically lay 4 eggs, an example of stabilizing selection. Larger clutches may result in malnourished chicks, while smaller catches clutches may result in no viable offspring. So নেচারে এমনই অদ্ভুত নিয়ম অনুযায়ী এখানে সিলেকশনটা হয়েছে যে যদি 4টের বেশি ডিম পারে তাহলে মানে the chicks will not survive, আর যদি 4টের কম ডিম পারে তাহলেও chicks will not survive. So this is the exact number of eggs they should lay, যার ফলে কি 4টেরই সার্ভাইভাল পসিবিলিটি বেড়ে যায়. So nature has selected this intermediate variation of the phenotype, and that is why it is called stabilizing selection, in which it favors the intermediate phenotypes. Second is directional selection. This is one of my favorite example. So directional selection কি হয় যে it favors one extreme phenotype causing a shift in allele frequency. So this is a classic example of peppered moth. So Biston betullaria. So Biston betullaria, there are two kind of varieties in this peppered moth. One is white and the other one is black. So before the industrial regulation, যখন pollution level অনেক কম ছিল, nature অনেক বেশি pristine ছিল, it was easy for this white variant of the moth to blend with the nature so that they can save themselves from the predators. Whereas the black variant, it was difficult for them to camouflage in the nature. So mostly কি হতো যে black variantটা বেশি predatorরা বেশি প্রে করতো, যে কোনো birds বা animals, they used to feed on the black variants, and the number of white variant was high at that time. But after the industrial regulation, যখন নেচারে ধীরে ধীরে পলিউশন বাড়তে শুরু করলো and the environment or tree barks starts to get darker in color, তখন these black variants, they used to get more favor, কারণ তাদের পক্ষে এটা ইজি হয়ে গেল to camouflage with their nature. So in a way, nature selected this black variant after the natural selection. So selection shifted towards a particular direction. যেখানে আগে এটা হয়তো ইকুয়াল ছিল না, হয়তো আগে এটা হোয়াইট ভ্যারিয়েন্টের দিকে বেশি ছিল, it shifted towards the black variant. And the last one is disruptive selection. So it favors the extreme phenotypes. What is called the extreme phenotype? So if there are like in nature, so if we look at this example, the last example of the rabbits, so if there are like 3 variants of rabbits, the black, the white, and the gray rabbit, so it will select the gray and the black rabbit, but not the white rabbit. So because in nature, the gray and black variant, they will have higher chance of survivability because they can easily blend in with the nature or they camouflage themselves in the nature. But the white variant, it is difficult for them to blend in with the nature. So they will be hunted more and they will be predated more and they will get extinct. So here the nature favors the extreme phenotypes, either the black or the gray, but not the white phenotypes. So this is how natural selection works. These 3 kind of natural selection, and the population demography or the genetic variation changes because of this kind of natural selection. And the last one is non-random mating. So Heidenberg, they propose that the population should be a random mating population. There should not be any kind of mate selection. But in real world, non-random mating exists. So females tend to select their mates based on some specific traits. So non-random mating involves individuals preferentially choosing mates based on some specific traits. So non-random mating can lead to changes in genotype frequencies and affect the distribution of genetic diversity within a population. So non-random mating বা mate selection এর ক্ষেত্রে যে দুটো কনসেপ্ট বা থিওরি রয়েছে, that is my favorite. That is, one is Jahabi's handicap rule and Fisher's runaway theory. So in that theory, they explain Jahabi's handicap principle suggests that females prefer males with survival reducing trait or handicap traits. So যদি আমরা পিককের এক্সাম্পলই ধরি, so পিককের যে secondary sexual characteristics, that is their tail, it is kind of handicap for them. Because of this large tail, it is not possible possible for them to fly or run from the predators. But still, this trait is selected by the females because it also suggests that having this kind of a handicap trait, the individual is still surviving in the wild. So that's why the females tend to select these males with longer tails. And fishes runaway theory supports Jahabi's handicapped model in a way that what happens that in a situation where the longer tail peacocks or the bright colorful tail peacocks are getting selected by the females, it becomes like a snowball effect, because in all the generation only this kind of males will be selected by the females, and this specific trait become fixed in the population so that these kind of males can be selected by the females. Here this is a classic example of these two theory. So if you see, there are like a short tail bird and a long tail bird. So short tail male birds, their genetic quality will be like good or bad, but they can survive in nature because of their short tail, because this is a cheap trait. But for long tail birds, they will be only alive if their genetic quality is good enough for their survival. But if the genetic quality is bad, they will be dead. So here is a graphical representation of this particular theory. So if there is like a short tail up to a typical length, then the survival advantage is high for the individuals. Then there is this second bracket where the tail length is increasing, but survival advantage and female choice is also increasing. And after this point, there will be like increased tail length, there will be female choice, but no survival advantage. So this kind of trait will be not selected in the nature, this kind of trait will be not selected, but a particular trait which has a prominent tail length and has a particular choice by female, this kind of trait will be fixed in the nature, so that particular this kind of male can pass on their genes to the next generation. So I believe all of you have understand these five concepts of Hardy-Weinberg theory, how Hardy-Weinberg theory works, and then I will come to the point of like population, gene pool, and genetic diversity. So what is a population? Population is a fundamental unit. Population is a group of interbreeding individuals that share a common geographic area. So একটা জিওগ্রাফিক এরিয়ার মধ্যে যখন একটা পার্টিকুলার স্পিসিসের অনেকগুলো ইন্ডিভিজুয়াল থাকবে এবং they will perform this random mating, and there will be interbreeding individuals and they will be producing viable offspring. তো তখন সেই particular geographic area with those particular species will be called as a population. And then what is gene pool? So the gene pool encompasses all the alleles present in a population. So all these individuals who are residing in that particular population and their genetic variation, what they are contributing to this population, will altogether called as a gene pool. And then there will be like genetic diversity. So what is genetic diversity? Genetic diversity refers to the variety of genetic material within a population. So as I have mentioned, all these individuals, the kind of genetic variation they have and the kind of genetic component they are introducing to the population will altogether be called genetic diversity. And the importance of genetic diversity. So the genetic diversity is essential for adaptation to the environment and disease resistance. So as I have mentioned before, the genetic diversity is high হবে, and তত বেশি there will be like chances of population to survive in the nature in the face of different kind of natural calamities or disease and anthropogenic effects. And genetic diversity higher বলতে যেটা বোঝায় সেটা হচ্ছে যে heterozygous component should be high. So there should be like always different kind of alleles, not like just a particular allele, not like all the dominant alleles or all the recessive alleles. It should have a proper balance between the dominant and recessive alleles, so the population can maintain heterozygosity and it can survive in the nature. So genetic diversity is everywhere. Genetic diversity exists between individuals. It can exist between population and between species. So when we talk about genetic diversity and conservation of wildlife, it's not just conserving one particular individual or one particular population, because we have to protect the biodiversity altogether. For that, we need to conserve all the species in a particular biodiversity, in a particular ecological area, so that the richness or the environment can be saved in that particular forest or in that particular habitat. So it is an overall concept to protect the genetic diversity. So low genetic diversity can lead to inbreeding depression, as I have mentioned before. So it not just to protect the individuals with same kind of genetic variation. So here you can see that it may be seen as a larger population, but it all has the same kind of individuals. So in this case, there will be like inbreeding. So inbreeding is basically means the mating between the same kind of individuals, and because of this inbreeding, only the particular type of alleles will be shared, and there will be no variation in the population. And if something happens, like any kind of natural calamity or disease spread, so the chance of surviving this population is very low. Whereas on the other hand, this population may seem smaller, but they have like different kind of individuals, so their genetic diversity is higher than this population, and because of this, their survival chance is much higher than this bigger population. So we have to understand the population. So first of all, we have to study the population for understanding the genetic diversity and inbreeding depression. তো প্রথমেই যখন আমরা কোনো ওয়াইল্ড লাইফ স্টাডি করি, we try to find out like what is the genetic diversity in that population is in inbreeding or not, এবং সেখানে কোনো রেয়ার অ্যালিলস আছে কিনা সেই পপুলেশনের মধ্যে, and then we try to formulate our strategy to safeguard this specific population. So this kind of information is important to know the genetic diversity and inbreeding depression of a population. So endangered species suffer from low genetic diversity. So আমরা যখনই wildlife conservation এর কথা বলি, we always try to focus on the endangered species. And we all know that based on IUCN category, there are different categories. So vulnerable, endangered, and critically endangered. So যখনই আমরা endangered species এর উপর কাজ করি, like for me, I have worked on red panda. So it is a group of species or species which have like low genetic diversity because of habitat fragmentation, urbanization, deforestation, agriculture. So already their habitat is fragmented, their population are kind of pocket population or isolated population. So there is high chance of inbreeding in isolated island populations, কারণ সেখানে number of individuals কম থাকে, number of mates কম থাকে, so mate selection happens between the similar kind of individuals. So এর ফলে কি হয় যে genetic diversity আরো লস হতে থাকে inbreeding এর জন্য, এবং the population tend to become more homogeneous, and their chances of getting survival in any kind of natural disaster or wildlife diseases is lesser. So genes for the future. So we have to protect genes for the future. So because climate change is forcing species to adapt to new conditions or move away, that is to get extinct. And high genetic diversity means there are genetic variants that might be suited to the new conditions. And higher genetic diversity provides a population with more tickets in the lottery. So if we want to protect a particular species or a group of species for the future generation, then we have to protect the overall genetic diversity of a population, of a habitat, of a forest, or of a particular biome. So we have this new concept called CBD, Convention on Biological Diversity, which is proposed in 1993 or 4, I guess. So CBD is a multilateral convention signed between different countries. So যেখানে মেইন এজেন্ডাই হচ্ছে to conserve the overall biodiversity based on genetics, and there should be like equal sharing of the genetic resources and sustainable use of genetic resources. So CBD is an old concept now, and we all know about the HE biodiversity targets. So HE biodiversity target set করা হয়েছিল for 2011, and then it is extended to 2014 or 15, but we are still lacking to protect this genetic diversity, because they have mentioned in HE targets like we have to protect a certain amount of genetic diversity in the world worldwide, but we are still continue to lose this genetic diversity now. So yeah, this is a concern for us, like how to protect the genetic diversity for the longer period of time, so that there can be like variation in the nature, not just for the survival of the wild, but for also our survival. Because if the genetic variation or genetic diversity tends to get lost in the nature, then it will affect us. So hopefully these concepts are clear now. So I have talked about so many things that inbreeding depression or genetic diversity, and but how to quantify this kind of genetic variation in a population or in wildlife. So যখন কোনো ওয়াইল্ড লাইফ আমরা স্টাডি করবো জেনেটিক দিক থেকে, তো কিভাবে করা শুরু করব? So for that we have different kind of molecular markers. So marker is a piece of DNA molecule that is associated with a certain trait of organism. সো আমরা এই যে প্রত্যেকটা ইন্ডিভিজুয়ালের মধ্যেই ডিএনএ রয়েছে, এবং ডিএনএ ইজ কনসিস্ট অফ ডিফারেন্ট কাইন্ড অফ দিস নিউক্লিয়ার ডিএনএ হ্যাজ ডিফারেন্ট জিনস। সো যদি একটা ডিএনএ হয়ে থাকে, সো দের উইল বি লাইক ডিফারেন্ট জিনস ফ্রম different region of this particular DNA. So what we try to do, we try to study this as many possible genes as possible for us, because there are like different kind of molecular marker available. So like mitochondrial DNA marker hote pare, nuclear DNA marker hote pare, আরএফএলপি করতে পারো, পিসিআর করতে পারো। সো বিভিন্ন প্রসেস রয়েছে, এবং বিভিন্ন প্রসেসে আমরা হয়তো একটা পার্টিকুলার ডিএনএ বা একটা পার্টিকুলার অর্গানিজমের ডিএনএ কিছুটা পোর্শনই হয়তো স্টাডি করতে পারি। বাট নাওয়াডেজ উই আর ওয়ার্কিং উইথ হোল জিনোম ডেটা। এনজিএস কনসেপ্টের ব্যাপারে তোমরা হয়তো জানো, যে নেক্সট জেনারেশন সিকোয়েন্সিং, যেখানে আমরা হোল জিনোম ডেটা নিয়ে কাজ করে থাকি। তো বাট দ্যাট ইজ অলসো লাইক ভেরি কস্টলি অ্যান্ড টাইম কনজিউমিং, হেভি ডেটা। সো যখন আমরা একটা লার্জ পপুলেশন নিয়ে কাজ করছি, বা একটা ফরেস্টের একটা পুরো ওয়াইল্ড লাইফ পপুলেশন নিয়ে কাজ করছি, so in that case it's not possible to study all the individuals with like whole genome markers. So what we do, we basically use the um, small microsatellite markers. So we started with allogymes, then RFLP or minisets, and nowadays we are working with PCR based markers like the microsets, which are most cost effective and easy to use, and it gives like most possible information that you need to conserve a population. So PCR based markers or SSR, simple sequence repeats, or microsatellites are these are like small markers, 18 to 25 base pair primers. So SSR polymorphisms are based on number of repeat units and hyper variable region. So যেটা বলছিলাম যে different organism of a same species, they have DNA and they have particular gene, but this gene component can be like different for different individuals, কারণ প্রত্যেকটা ইন্ডিভিজুয়ালের জেনেটিক কম্পোনেন্ট আলাদা হবে। সে জিন যদিও সেম হয়ে থাকুক না কেন, তার কিছু না কিছু ভ্যারিয়েশন সেখানে থাকবে। সো we should select some kind of marker, genetic marker, which can reflect this variation in different individuals of the same species. So যখন microsatellite marker use করা হয়, microsatellite markers are hyper variable. Hyper variable meaning হচ্ছে এটা যে they can capture different kind of variability in the genes of different individuals. So এই particular যদি gene 1 এর এখানে কিছু ভ্যারিয়েশন থাকে, and I use this particular microsatellite marker for these two individuals, individual 1 and 2. So this particular marker, this particular SSR or STR marker can capture this hyper variable region and can tell me about the difference in the genetic variation between these two individuals. So that is why we try to use STR markers, because this is easy to use, and this is also like cost effective. And when we try to study a big population, or when we try to understand the number of individuals in the population, in that case microsatellites are most cost effective. So after the PCR, what we get like this is the gel electrophoresis, যেখানে বিভিন্ন জিনের আমরা ডিএনএ ফ্র্যাগমেন্টের লেন্থ অনুযায়ী আইডেন্টিফাই করতে পারি যে কোন কোন জিনগুলো রয়েছে বা কোন কোন জিনগুলো are getting amplified in particular PCRs, and this is how it looks on the capillary electrophoresis. So basically যখন আমরা STR marker use করি, we try to find out the allelic frequency of a particular gene, so that we can use it for further analysis of the genetic variation of the species. Yeah, so now let's come to the applications of conservation genetic techniques. So conservation genetics এ যে basic fundamental concepts গুলো আমি ডিসকাস করলাম এতক্ষণ ধরে, যে what is Hardy-Weinberg theory, how Hardy-Weinberg theory help us to understand about the genetic diversity and the genetic variation that is happening in the population, and how we can incorporate this concept or this principle to study the wildlife genetics using different kind of molecular markers. So that is the application part of the conservation genetic techniques. So we can do genetic monitoring and for the conservation of the wildlife. And the second point I'm going to discuss, that will be like wildlife forensic science. Okay, so now before going further, so everybody is clear, or am I boring or something? All of you can hear me?
Yes, you are perfectly audible. Please carry on.
Okay, okay, thank you. So first, I'm going to discuss about my PhD work, that is the population genetics of red panda in Arunachal Pradesh. So as I was mentioning that my PhD work is like very special to me. It's not just about the species, it's about the overall experiences that I had during my 6 years of PhD. So this is the species distribution map of red panda by IUCN. So this is a transboundary species. It is found in 5 range countries, that is Nepal, Bhutan, China, northern part of Myanmar, and obviously in India. So and in India, it is like found in Sikkim and northern part of West Bengal and in Arunachal Pradesh. And there is also an unconfirmed population from Meghalaya. So I have worked in both Sikkim and Arunachal Pradesh for trying to understand the genetic component of red panda and to suggest the mitigation for different kind of problems they are facing. But here I am going to discuss the results of Arunachal Pradesh, what I have worked there. So basically, Arunachal Pradesh is a very interesting landscape in a way because it's in the northernmost part, most in the most remote area in the northeast part of India, because it is totally cut off like from the mainland in a way that because of its rough terrain, there is like less accessibility during the monsoon. যখন আসামে flood আসে, this landscape of Arunachal becomes like cut off from like rest of the India, because আসামে flood এলে এখানে যাওয়া is getting impossible. And one important thing is that it is a transboundary region, so it shares border with Bhutan in the west, Myanmar in the east, and China in the north. And because of that, there is always a high chance of illegal wildlife trade in this landscape. It's because it's present in the conjugation of 2 very important biodiversity hotspots. So one is the Eastern Himalayan biodiversity hotspot, and the other one is the Indo-Burmese biodiversity hotspot. So যেহেতু it's present in between these 2 biodiversity hotspot, so the species richness is enormous. I mean, there are like different kind of species, and so many species are still to discover in this landscape. So yeah, and it was not difficult to work in this landscape because of this rough terrain. So as you can see in the elevational gradient, the elevation is up to 6,600 meter. And red panda is a species of from like 2,000 meter to 4,000 meter. So it is difficult to reach the habitat of the species, because in the high altitude region of Arunachal Pradesh, there is like very less accessibility, no road connectivity or nothing else. And also it is a transboundary region, so there is always a high military restrictions in some parts of the region. So it was not possible to go to all the areas and collect samples. So what I have done, I have tried to follow a process called representative sampling. So shekhatra ami I tried to collect samples from each part of the landscape. So I collected samples from the western part of Arunachal Pradesh, the central part, and from the eastern part of Arunachal Pradesh. So we have done a line transect survey and 5 by 5 kilometer grid, and we collected possible samples of red panda from the wild. And this is the basic methodological workflow. So first of all, we do field work in the species distribution range areas. And then we try to collect the fecal samples and the ecological data. So these are important. Karon, as I have mentioned, the endangered or critically endangered species নিয়ে কাজ করার সময়, it is not possible to get permission to collect blood sample or tissue sample from the animals. And it is not even possible to collect blood samples for red panda, because red panda দেখাই is kind of next to impossible Eastern Himalayas, and তারপর তাকে capture করা blood sample, and it's not permissible also. So the best way to have genetic data from this kind of elusive species is to collect the fecal sample and then extract DNA from fecal samples. So এটাকে আমরা বলে থাকি নন ইনভেসিভ প্রসেস অফ ডিএনএ এক্সট্রাকশন। তো নন ইনভেসিভ প্রসেসে আমরা ফিকাল স্যাম্পল কালেক্ট করি এবং ফিকাল স্যাম্পলের যদি উপরের লেয়ারটা দেখি, দ্যাট ইজ কল্ড দি এপিথেলিয়াল লেয়ার। সো ইট হ্যাজ সাম কাইন্ড অফ শেডেড টিস্যু। সো when the animals defecate, so some tissue layers also shed with the fecal matters, and we scrap out this upper layer of the fecal samples, that is called the epithelial layers, and this upper layer of the fecal samples that consists the DNA of that particular species. So we bring the fecal samples to the lab and we extract DNA from these fecal samples and we go for the genetic analysis. And on the other hand, we also collect the ecological data. Ecological data means the location points, GPS location points where we are collecting the samples, then the altitude or elevation, the kind of forest type, the slope or aspect from where we are collecting the samples. So these are kind of ecological data we also collect, and we use this ecological data for building species distribution model. So for my PhD, I also have conducted questionnaire survey to understand the threat to the species and how we can mitigate these threats for the conservation of the species. So after collection of samples, you can see a brief overview of how we process the samples. So first of all, genetic, gDNA extraction. Then we process this DNA for PCR with microsatellite markers that I have mentioned earlier. There are like microsatellite markers we use to understand the nuclear component of the species. And then these PCR products are used for generating genotype or sequencing data. And this genotype and sequencing data are finally used for analysis to understand the genetic diversity and other genetic components of the species. So my first findings for red panda was that red panda exists in two phylogenetic species. So earlier it was believed that red panda is a monotypic species of the family Ailuridae. But after this first ever genetic study of red panda from India, I have found that it's not the true concept. Red panda actually exists in two phylogenetic species, and the separation for divergence between these two species is the Siang River or the Yalujangbu River or the Brahmaputra River. As we may know of this river, it originates from the Tibet and it flows from China, then to Arunachal Pradesh, and then it enters Assam as Brahmaputra River. So I have collected samples from both sides of the river in Arunachal Pradesh, in Sikkim, and in the eastern Arunachal Pradesh, I have collected samples from Dibang. And then we have the NCBI database from where I have also gathered sequences of red panda from earlier studies. And when I analyzed all these samples together, I found something very interesting, that all the samples which are on the western side of the river, they clustered together, and all the samples which are on the eastern side of the river, they clustered separately. So based on this, I have hypothesized that there are like two different phylogenetic species of red panda. One is the Himalayan red panda and the other one is the Chinese red panda. And this is also this fact is also now prominent, that India is home to both the phylogenetic species of red panda, because the eastern population of red panda in the eastern Arunachal Pradesh, in Dibang Valley particularly, is of Chinese variant of red panda. So this study was first proposed by a group of Chinese researchers in 2020. So they have this first publication with the whole genome dataset, but তাদের publication এটা lacking ছিল যে they didn't get samples from Bhutan and India. So they just bring out this concept based on the samples collected from Nepal and China, and they have mentioned that red panda exists in two phylogenetic species. So we first didn't agree with this concept, but now after doing our own analysis, we kind of agree to the fact that red panda actually exists in two phylogenetic species. And here you can also see the phylogenetic tree. So this is something we build with the mitochondrial DNA markers data, যেটাকে আমরা sequencing data বলে থাকি। তো different sequencing data যখন আমরা analysis করি from all the individuals, and we can find the patterns in phylogenetic tree, and you can see like how this pattern is. Like here we can see that there are like two different clades for red panda. The clade 1 belongs to Ailurus fulgens, that is the Himalayan red panda, and the clade 2 belongs to Ailurus tiny, that is the Chinese red panda. And these red dots, they belongs to the samples collected from the Dibang Valley National Park of Arunachal Pradesh, which is in the eastern side of the Siang or Brahmaputra River. So based on the phylogenetic tree, it is also evident that yes, these two species are quite separated phylogenetically, and they can be like accepted as two phylogenetic species. And these two species, I have found that they have diverged at around 0.30 million years ago. So মোটামুটি 3 lakh বছর আগে this divergence have happened. So maybe it's happened during the late Pleistocene time. So maybe there are like because of the glaciation and repeated cycle of glaciation and deglaciation. So these kind of natural events may have caused this separation of the two species. So then, as I was mentioning, we can do species distribution model with the kind of ecological data we have. So as I mentioned that I have collected samples from different parts of the landscape. So we had the location data from different parts, and then we collected different kind of climatic variables, then different kind of topographic variables like elevation, slope, aspect, and different kind of forest types like we have this online database from where we can get this different kind of forest type data also. Then anthropogenic data sets like urban areas or the road connectivity networks. So this kind of data we can gather from online sources and from the ground survey also, and then we can use these data sources with the location points data we have to find out the suitable habitat for the species in the landscape. সো যারা এই কনসেপ্টটা বোঝানো তাদেরকে একটু সিম্পল ভাবে যদি বোঝাতে হয়, তো যদি রেড পান্ডার মতো বা এরকম কোনো স্মলার ম্যামলস আমরা কাজ করি which are not easy to find in the wild, it is not possible to survey all the areas in the study landscape to find out where the species can be found. So সেক্ষেত্রে species distribution model is a very fine concept. তো যেখানে আমরা কি করি যে we go our to the field and do our field survey from possible regions যেখানে আমরা হয়তো species এর দেখা পেতে পারি বা we can find their signs. For me it was like fecal samples. So যখন আমরা representative sample থাকে from different parts of this landscape, and then I have the different kind of climatic variables or topographic variables. So what this function basically do, so if we have a location point from here, so the species distribution model, what it does, it extract the bioclimatic or topographic variables around these location points to find out what is or what type of environment is suitable for this species, and then it extrapolate it for those areas where the species points are not available, but it tries to find out if similar kind of environmental habitat is present there or not. So যদি সেখানেও similar kind of environmental habitat present থেকে থাকে, তাহলে it will predict that this area can also be a suitable area for the species based on the similar environmental conditions and the availability of resources. This area can be also possible for the species to habituate or reside. So this is how the habitat suitability model works. So what I have found in this case that most of the suitable areas for red panda actually exist outside the protected area network. So here if you see, like only 0.5% area of the suitable habitat are inside the protected area network, and almost 14% is outside. So এত বড় ল্যান্ডস্কেপের মাত্র 14.5% is suitable for the species. সেটাই একটা বড় কনসার্ন আমাদের জন্য যে এই স্পিসিসটাকে এই ল্যান্ডস্কেপে প্রোটেক্ট করা বা কনজার্ভ করা is important. And the second part is that this habitat is outside the protected area networks. So we should have a protected area network যেখানে কিনা these animals can be protected properly. But for landscape like Arunachal Pradesh, it is not possible because this is a tribal area, and all these tribal communities have their own, different kind of their lands, and government cannot always, interfere with their land use types. So, and these tribal communities, they are also like hunters, gatherers. So they still practice different kind of hunting methods as their livelihood, and there is a shift towards for these communities towards wildlife trade or illegal wildlife crime, because there is as I have mentioned, that there is a high demand of wildlife products from this region, because this is a biodiversity hotspot region. They have like enormous species diversity, and as this is present in the border with China and Myanmar, so in the Southeast Asia there is a high demand of wildlife products from this part, and so that's why like pangolins, red pandas, musk deer, black bear, these all animals are like hunted from this region, and exported to different parts of Southeast Asia. So now we understand what are the problems, so how we can conserve the species in the landscape. So first of all, we need to understand the genetic diversity or genetic component, as I have mentioned previously also. So that is very important. So we have to understand if the population in Arunachal Pradesh are genetically fit or not. So for that, I have performed different kind of genetic analysis. So in a way, basic. So this is called structure analysis. Each bar represents here a single individual. So if we follow this, so we can see like there are at least 4 different kind of population. So each color represent a different population. So blue, yellow, green, and red. So 4 color means 4 different kind of population exist in the total landscape of Arunachal Pradesh. Then there is this scale from 0 to 1. এই স্কেলটার মিনিং হচ্ছে এটাই যে যদি কোনো একটা বার or particular একটা individual is more than 80% in height, then it can be said that this particular individual is assigned to a particular population. So, and if it is like below the 80% range, then it means and have like different color shades, তার মানে হচ্ছে যে that is an admixed individual and it has like sharing of genes from different populations. So for example, if we try to understand, if we look into this kind of this individual here, yeah, so this individual, red individual, so it has more than 80% of a single color. So this can be said that this individual is assigned to a particular population. Whereas if we look into this individual particular, so this has a different shades of yellow, blue, and green, yellow, blue, and red. So this is an admixed individual which have like genetic components shared from this population and this population and this population. So this is an admixed individual. So basically what it explains that the population in Arunachal Pradesh, it's exists in a meta population framework. Meta population is a population where there is like gene flow between the population. There is like sharing of individuals between the population. So it means the population is sustaining in the wild. There are like not clear-cut signature of like closed population or like pocket population. Rather, the population are sharing, having shared genes among them. So it is based on this we can plan to protect these species on the concepts of populations. And one interesting fact is that this DB population here, which is the Dibang Valley population, which is in the eastern side of the landscape, which I have mentioned before is maybe belongs to the Chinese red panda population. Here also you can see some level of admixing with these populations. And the signature of this population, yellow population, can be found here and here also. So now this is like really in question, like if there is like really 2 phylogenetic species or not, and this need to be this need to have further research to understand this concept of 2 phylogenetic species in red panda. So yeah, based on this genetic data, we can also have something like this. This is called genetic divergence map. So genetic divergence is basically the genetic distance between 2 populations. So for the ease of understand, if I try to explain what is genetic distance between 2 individuals, between 2 populations. So suppose these are like 3 different populations. So maybe these 2 population has good amount of gene sharing or kind of like good amount of gene flow from one to another. So they will have less genetic distance or less genetic divergence. Whereas these populations are like far behind. So this population may not have like any kind of gene flow or genetic connectivity with these 2 populations. So it will have a high genetic distance with these 2 populations. So this is the concept of genetic distance or genetic divergence. So if we try to observe it on the map, we can see that these red areas, this is this particular scale of genetic divergence. So if we try to find out or understand the genetic distance between different kind of populations, so we'll see like the populations present on this and this side of this landscape, they have a high genetic distance in this region. And in this portion also, the genetic divergence is high. And this portion also, the genetic divergence is very high because the color is totally red in these particular areas. So now the natural question is, what might be the reason behind this high genetic divergence in these regions, and that is the beauty of the landscape genetics, or conservation genetics study. So if we see, like, there are like different kind of mountain passes are present, and these are the different kind of river networks are present in these landscapes, and based on this, we can say, or we can predict that it may be possible that it is because of this different river network in the landscape, or these different high mountain passes in the landscape. There is like no movement between the populations, or no gene flow between the populations, and that is why the genetic divergence or genetic distances are high in these particular areas. So based on this, if we try to find out the gene flow or connectivity network, so this is how it looks like, how the gene flow works in the landscape. So for red panda, this is it can be observed that in the western part and in the central part, the gene flow or connectivity is high. But as we go to the eastern side of the landscape, the gene flow or connectivity, it reduces from the western and central part. It is maybe because of these high river networks and the high mountain passes that I have discussed on the previous slide also. So this is how landscape genetics work, or gene flow works. So these are the approaches we used in population genetics analysis, and based on this, we suggest the management strategies. Here we can find the different kind of habitat core areas, and the areas which have like high barrier. So this is the scale for barrier, low to high. So these blue regions have like high barrier for gene flow, so the gene flow are like less in these particular regions. This is the habitat core value. So the lesser the value, the less important the habitat core areas are. The higher the values, the high important the core variables, core habitats are. So from this we can understand that in this particular landscape of Arunachal Pradesh, these particular two habitat cores are the most important habitats. So these all findings are based on genetic data and the habitat suitability map that I have discussed. So this is basically how genetic data helps us to understand not only the genetic structure or the genetic component of a species, it also helps us to understand how this is spatially reflect the population's condition in the landscape. So as we can see here, like, we can find out the important core habitat and the areas with less gene flow, and based on this we can propose our conservation strategies. Just like this. So we can see the these are the LCP, or the least cost pathways, that represents the connectivity. So these cost pathways which have lesser values are non-effective cost pathways, whereas these red color pathways which have the higher values, they are the most effective pathways. So if we see, like, here the red color pathways can be seen, and here also, so we can conclude that these are the particular areas we need to conserve in this landscape, and these are the particular habitat patches we need to conserve in the landscape, so that a long-term viable population can sustain in this area. So this is how we propose conservation strategies based on the genetic monitoring of a species in the landscape. And for this, it may not be possible to establish a protected area for a single species, but we can go for like community conservation approach, where we can talk with the local communities and make them agree to have a kind of their own protected area or community conservation area, in particular these areas, so that they can protect this landscape for the species conservation. So yeah, this is a brief of work of my PhD, which explains how genetic monitoring works for population conservation or wildlife conservation in a particular landscape. So now I'm going to discuss few of my colleagues' work on the population genetics aspects. So first of all, I should explain this work of Sela Makak, because this is very interesting. So Dr. Abhijit Ghosh, আমার colleague, এবং আমরা একসাথেই PhD করা start করেছিলাম. So he was working on Arunachal macaque and I was working on red panda. তো অভিজিৎ দার field site ছিল mainly তাওয়াং and ওয়েস্ট কামেং ডিস্ট্রিক্টে অরুণাচল প্রদেশের, and আমি মোটামুটি গোটা অরুণাচল প্রদেশেই কাজ করছিলাম. So when he started working on Arunachal macaque itself is a very new species of primate. It is discovered by Dr. Anindya Sinha in 2010 or 9 or 10, I guess, probably. So it itself is a very new species, and there has been very less work on this species altogether. So he started his work in Tawang district, and he collected his sample from Tawang and then from West Kameng, and then he also got some samples from the central part of Arunachal Pradesh. And he found something very interesting. He looked into the individuals from different district, like in Tawang, and these are the individuals from West Kameng district, and you can clearly see the differences in the individuals. The individuals from the Tawang, they have like thick furs, their coloration is different, their facial features are kind of different. Whereas if you see the individuals from the West Kameng district, so this is the Tawang district, and this is the West Kameng district. So the individuals of West Kameng districts looks totally different from these individuals. Their fur is not that much thick. Their facial features is somewhat different. So he got very curious কি এরকমটা কি করে হচ্ছে যে both are like same species described as Arunachal macaque কিন্তু এদের দুজনকে দেখতে আলাদা. So he started his genetic analysis, and he found that all the samples he have collected, they are clustering separately. So if you see here, these green circles, this represents the samples from Tawang district, whereas these blue ones represent the samples from West Kameng district, and these yellow ones represent the samples from the central Arunachal Pradesh. And these lines, this represents the number of mutations that has been happened between these populations. So this is like huge number of mutation, huge. So 28 or 25 number of mutation is considered very huge number of mutation between two populations to become a new species. So it was really an interesting finding. Then he started to look into other aspects of genetic, and as I have mentioned earlier, also this is the genetic structure of a population or wild population. So এখানে যদি দেখা যায় যে red panda যেরকম genetic structure ছিল এটার মধ্যে কিন্তু totally different. We for red panda we had like different individuals with different colors. So তার মানে ছিল যে সেখানে gene flow বা connectivity ছিল, and there were like exchange of genetic components between the populations. But here we can see like Tawang এর যে individuals গুলো, they assigned to a single color, only green color, and West Kameng individuals গুলো are of like only one single color, and they are like more than 80% in a single color. So তার মানে হচ্ছে যে এই individuals গুলো particularly assigned to West Kameng district, and these are assigned to Tawang district. Even on all the other genetic um, analysis, he has found the same results here also, and based on these results, he also done this phylogenetic tree analysis. So Macaca munzala is Arunachal macaque, and Macaca selai is the Sela macaque. So if you also see on the phylogenetic tree here, so তাহলে দেখা যাচ্ছে যে Macaca selai, they are forming a different clade from all the different macaque species. So Macaca radiata, Leucogenes, tibetana, so these are all different macaque species that we already know about, and this is the Arunachal macaque, and this is the Sela macaque. So on the phylogenetic tree also he has found some differences between these two group of population from Tawang and West Kameng, and based on this he proposed that these two are totally different phylogenetic species, and he claimed that this cause of separation between these two species is because of the high mountain Sela Pass in this region. So we all know that Sela Pass is more than 3,500 meter or 4,000 meter in altitude. So, and it has no vegetation or nothing in this area. So it was not possible for the individuals to cover this landscape, and it was a perfect explanation for the species divergence in this region. So this is the most beautiful aspect of wildlife conservation genetics study, যে যেখানে based on genetic data আমরা অনেক কিছু conclude করতে পারি যেটা হয়তো খালি চোখে দেখে বোঝা যায় না বা নর্মাল ইকোলজিক্যাল স্টাডি করে হয়তো বোঝা যায় না, কিন্তু genetic data can explain that differences. So it was a very interesting study by him, and he explained this new species in 2021, I guess. He published this paper, and it's now totally a new avenue to work on. Yeah, so the second most interesting discovery by ZSI or by our group in Arunachal Pradesh was the discovery of white chick macaque. And এই ইনসিডেন্টটা আমার সাথেই হয়েছিল। সো যেরকমটা আমি বলছিলাম যে আই and Dr. Abhijit Ghosh, we used to work together in Arunachal Pradesh. So ওর কাজ মোটামুটি তাওয়াং এবং ওয়েস্ট কামেং ডিস্ট্রিক্টেই কনসেন্ট্রেটেড ছিল, and I used to go for field survey in all over Arunachal Pradesh. So he has mentioned to me, like, if you're going to the different parts of Arunachal Pradesh, you collect macaque samples, whatever macaque samples you get, you bring it for me. So আমি ভেবেছিলাম ঠিক আছে, যে কোনো বাঁদরের স্যাম্পল পেলে আমি নিয়ে যাবো দাদার জন্য. So I was doing my field survey in সেন্ট্রাল অরুণাচল প্রদেশ সিয়াং ডিস্ট্রিক্টে। সো সেখানে আমি বাঁদরের স্যাম্পল খুঁজছিলাম, আমি একটা গ্রামে ছিলাম, অ্যান্ড দেন আই কাম টু নো অ্যাবাউট আ লোকাল হু ইজ was holding a macaque child. So তাদের বাড়িতে গিয়ে আমি জুভেনাইল ইন্ডিভিজুয়ালটা দেখতে পাই বাঁদরের। সো আমার তখনও এতটা আইডিয়া ছিল না বাঁদরের ব্যাপারে। সো আই থট ইট মে বি পসিবল ইটস অরুণাচল মাকাক। সো আমি এই ইন্ডিভিজুয়ালটার ফিকাল স্যাম্পল কালেক্ট করি। দেন আই ওয়াজ ইন দিবাং ডিস্ট্রিক্ট অলসো, অ্যান্ড সেখানে আমাদের ক্যামেরা ট্যাপে এই ইন্ডিভিজুয়ালটার ছবি ধরা পড়ে, অ্যান্ড লেটার অন দিস প্লেস উই অলসো কালেক্টেড দি ফিকাল স্যাম্পল ফর দিস ইন্ডিভিজুয়াল. So then he did, Dr. Abhijit, he did some very good analysis, genetic analysis. And here you can see this is the sample I have collected from West Siam, the baby monkey. And this is the sample collected from Dibang Valley. And these two are like totally clustering in this particular group. These are like the different group of macaques, but these particular samples are clustering together, and he has identified it as Macaca leucogenes, that is the white-cheeked macaque. So it was not at all a planned work, but it was just something we discovered luckily or certainly. And this is the basic map of the species. So earlier it was believed that this was the previous range of the white-cheeked macaque. So a blue region totally, it's in Tibet or in China. So প্রথমে ভাবা হতো যে শুধুমাত্র এই পার্টিকুলার রিজনেই হোয়াইট চিক মাকাক পাওয়া যায়, বাট দেন উই কালেক্টেড স্যাম্পল ফ্রম হিয়ার দিবাং অ্যান্ড ফ্রম হিয়ার that is in central Arunachal Pradesh, and based on this we proposed this new extended range for the white-cheeked macaque. And this was the first discovery by ZSI and by our group that explains that India is also home to a new macaque species that was previously not known to be in India, that is white-cheeked macaque, and this is now also listed in our Wildlife Protection Act amendment. So yeah, this was also a great finding by us. And this is like very new, the discovery of Indo-Burmese pangolin from northeast India. This is also a great story. Like it was when I was in ZSI at that time, 2022-23 সালে. So ZSI is a premium institute, so we also work on wildlife forensic. তো যেখানে যত রকমের wildlife product seize করা হয়, they send it us to ZSI to do the genetic analysis and to find out which animal species it is. So pangolin being a most trafficked species, so always we get samples of pangolins, and our job is to find out that if the samples contain is of like Indian pangolin or Chinese pangolin. So for pangolin, it is like there are like in total 8 species of pangolin. তার মধ্যে 4টে হচ্ছে আফ্রিকান প্যাঙ্গোলিনের স্পিসিস, যেগুলো পুরোপুরি আফ্রিকান কন্টিনেন্টে পাওয়া যায়, অ্যান্ড 4টে হচ্ছে এশিয়ান প্যাঙ্গোলিন স্পিসিস, যেগুলো এশিয়ান কন্টিনেন্টে পাওয়া যায়. সো দিস 4 এশিয়ান প্যাঙ্গোলিন স্পিসিস আর দি ইন্ডিয়ান প্যাঙ্গোলিন, চাইনিজ প্যাঙ্গোলিন, জাভান প্যাঙ্গোলিন অ্যান্ড ফিলিপাইন প্যাঙ্গোলিন. তো ইন্ডিয়াতে যে দুটো স্পিসিস পাওয়া যায় সেটা হচ্ছে ইন্ডিয়ান প্যাঙ্গোলিন অ্যান্ড চাইনিজ প্যাঙ্গোলিন. সো চাইনিজ প্যাঙ্গোলিন পাওয়া যায় হচ্ছে শুধুমাত্র ইন্ডিয়ান নর্থ ইস্ট রিজনে, দ্যাট ইজ ইন দার্জিলিং, সিকিম, and Arunachal Pradesh, Assam. So this part has the distribution of Chinese pangolin in India, and in the rest of the India there is only Indian pangolin. So আমাদের জানা ছিল যে যদি কোনো কনসাইনমেন্ট এসে থাকে প্যাঙ্গোলিনের, so it might be like Indian pangolin or Chinese pangolin. So we did our normal analysis, but we found something very different. So this time the few samples we received, so these are the few samples, they were like clustering all together differently, not with the Chinese pangolin, that is Manis pentadactyla, and not with the Indian pangolin, that is Manis crassicurata, and not with other pangolin species, that is Sumatran pangolin or the Philippine pangolin or the African species of pangolins. So we started to wonder like what this can be, and we also tried to find out the divergence time of this clade, and it was like quite long before. It's 3.4 million years ago. Almost 35 lakhs year before this divergence has happened. So and this divergence has happened from the Chinese pangolin. So এখানে দেখা যাচ্ছে যে Manis pentadactyla, that is the Chinese pangolin, and this particular clade is diverged from this clade here. So Chinese pangolin থেকে এই cladeটা diverge করে এই নতুন cladeটা তৈরি করেছে about 35 lakh years ago. So that is like a long time back. So if it is like diverged before this much time, so it has to be something like a different new species. So it diverged in the area of Pliocene. So in the Pliocene, about 35 lakh years ago, this new clade has diverged. So we tried to find out what might be the reason for this divergence, and then we went for this modeling aspect, which is called paleo dam modeling. So if you see, this is the Paleodem model of 5 million years ago. So আজ থেকে 50 লাখ বছর আগে নর্থ ইস্ট ইন্ডিয়া বা নর্থ ইস্ট সাউথ ইস্ট এশিয়া দেখতে অনেকটা এরকম ছিল এলিভেশন গ্রেডিয়েন্ট অনুযায়ী। So there are like lower region which are like as low as like 3,900 meter, and there are like higher regions which are about 6,600 meter. But if we see the present elevational model of this particular area, then we can see that the higher elevation has become like 8,600 meter in this particular region. So there is a shift in the elevational gradient from 5 million years ago to the current situation. The 50 লাখ বছর আগে এখানে মাউন্টেনের হাইট অতটা বেশি ছিল না, বাট 50 লাখ বছর বাদে মাউন্টেনের হাইট অলমোস্ট 2000 মিটার বেড়ে গেছে, which is a huge margin. So it is possible that during this time period 35 35 lakh years ago something has happened যেখানে মাউন্টেনের হাইট বাড়ছিল এবং এখানে ল্যান্ডস্কেপ ফিচার্স গুলো চেঞ্জেস হচ্ছিল। So it might be possible a particular population get trapped in some area and it has become a new population, or it has diverged from the previous population and become the new species called the which we have named as Indo-Burmese pangolin. So this is like morphologically we tried to explain. So this is the lateral view of the Indo-Burmese pangolin, and this is for Chinese pangolin. So you can see like there is a difference in color and the scale patterns. Here also this is the scale, dorsal view of the Indo-Burmese pangolin, and this is for the Chinese pangolin. And there is a clear-cut differentiation in color and scale size, scales pattern. So based on this genetic data and the morphological data, this we have explained from ZSI the new more species, the Indo-Burmese pangolin, and this is very recent. This paper is just published in 2025 only, just a few weeks ago. Okay, so that was all about the approaches in wildlife monitoring using the approaches of conservation genetics. And now I will focus on the concept of wildlife forensic, because this is very important, and this concept of population genetics can help us to work on the field of wildlife forensic. So what is wildlife forensics? Wildlife forensic is basically a approach, or is a course of science in which we are concerned with the application of scientific analysis to support the wildlife law enforcement. So as I have mentioned earlier also, that the wildlife crime is a very prominent crime, and illegal wildlife market is very high. So there is always a high demand of wildlife products, and the wildlife are being killed because of this profit. And there are like different laws, but you have to prove someone guilty in front of the law so that they can be convicted. And for that, wildlife forensic is a good tool to use to prove the evidences. So yeah, so as I was mentioning, like wildlife crime, it is a global crisis, because if you see, this comes third highest profited or highest market, illegal market, just after cocaine and heroin, that is drugs and firearms. So it is a $53 billion business, illegal business, which includes like wildlife trade, illegal fishing, and illegal timber collection. So this all together combines around 53 billion, which is like second or third after the drugs and firearms. So you can imagine like how big this wildlife market is, and why the wildlife is still in this much threat in today's scenario also. So this is the global wildlife crime data. This is up to like 2017. You can see that from 2010 to 2017 only, the crime rate has doubled almost. And these are the most hunted species in the wild. So elephant, then pangolins, then rhino, and then there are like reptiles and big cats. So all of these wildlife are being hunted because of their demand in the wildlife trade. And this is you can see like how this graph has risen for different species and the hunting pressure. So pangolin is still the like most hunted species till 2017 and 18 also. Elephant hunting has somewhat reduced but is still in the threat list. Rhino hunting is increasing, tiger hunting is also increasing. So this is a specific concern for the wildlife conservationist or conservationer like us to protect this kind of species, and for this we need proper law and implementation of those laws. So if we try to understand the wildlife crime in India, so we can see that most of this wildlife crime is related to pangolins and the big cats and elephants. So these are the species which are involved in wildlife crime in India. So you can see different reports are being published on the wildlife crime from India, but unfortunately this is not being highlighted by the mainstream media, which is unfortunate. And we don't talk about this kind of wildlife crime in India, and that is why there is less awareness among the people about wildlife crime in India also. So why this wildlife crime is an attraction to the criminals? So as I have mentioned, it is the third profitable illegal trade market in the world, just after drugs and firearms. So but there is a less risk associated with it. So if you get caught for smuggling arms or drugs, you can go for a long time in jail. But for wildlife, if you get arrested for committing any wildlife crime, then you will not be punished for a longer period of time. And then it is difficult to identify. Identification is complicated of the particular wildlife product you are smuggling. It is because after killing the animal, you may have like different body parts of the species, and it is different and very difficult to identify those body parts. I mean, if it is belonging to Schedule 1 or Schedule 2, because in Indian Constitution there are like different punishment rule for killing different scheduled species. So obviously Schedule 1 species के यदि केउ poaching करे तार penalty बेशी হবে compared to Schedule 4. But the identification of this species is complicated. Then again, the conviction or penalties are far more lenient. It is not that much compared to like firearms or like murder or anything. So people get to get away easily, and fines often disproportionately less than the commodity values. So যদি একটা হাতির দাঁত বিক্রি করে 2 লাখ টাকা পাওয়া যায়, so একটা হাতি মেরে হয়তো তোমাকে 10,000 টাকা ফাইন করতে হবে. So obviously the crime is profitable. Wildlife illegal trade or crime is profitable, and it has less risk associated with it. So the criminals get already attracted to this kind of wildlife trade. So the low prosecution rate is a problem for our system. So if we cannot identify the wildlife articles that were being poached or that were being smuggled, then there cannot be a proper jurisdiction or cannot be a proper conviction to the criminals. So it is very important to identify the wildlife products associated with the wildlife crime. So this is the notorious case or the infamous case of Sangsar Chand, which is like still horrifying, and it still gives me goosebumps to discuss this. Like in 2004, Rajasthaner Sangsar Chanদের family থেকে police seized করে প্রায় 40 tiger skin and 400 leopard skin. And this was in between, like in 11 months, they have killed this many tigers and leopards. And in 2006, CBI get to know that Sangsar Chand has killed like more than 400 tigers, 470, and more than 2,000 leopards alone to supply these products to China, Nepal, and Tibet. So this was the scenario in India, and this is still somewhat maybe the same also right now, how wildlife crime is still happening in India, and what is the extent or the scale of this kind of wildlife illegal wildlife crime in India. And this is the the highlighted case, but there are like so many different cases which are all of like significant magnitude of this one. So yeah, this is very unfortunate that we don't have like proper control situation on this wildlife crime. So we need to control, or we need to have a proper system to control this wildlife crime in India. So this was the pathetic scenarios that has been captured from Sangsar Chand's family. So in wildlife forensic, what are the challenges that we face? So suppose we get samples like body parts which are like roasted animal body parts, how you would identify this species. Then there can be like a crime has happened in a particular forest or in a particular area, but you didn't get any body remains, but you only get the soil soaked with blood. So how you will find out like which animal has been killed in that particular area. Then you can have like raw meats or cooked meats, or like this kind of animal product, and you have no way to find out which animals. So if you want to bring a person to conviction, you have to identify these articles as they belong to any wildlife products or not, or they belong to any schedule 1 species or not, and based on that, that person will be convicted. So it is very important to have a particular system to identify this kind of products. So ZSI is contributing to wildlife forensic, and we have our labs where we do different kind of genetic analysis to find out these wildlife products. So ZSI 16 regional center with the Pasta regional center, we have DNA barcoding facilities, and we have the the state of art facility in our headquarters in Kolkata, and there's this new lab where we have all kind of facilities to study the wildlife forensic. So this is kind of the cases that we have received till 2022, about 565 cases in ZSI. And these are the number of cases we received from different state. So you can see like most number of cases are received from West Bengal. This is probably because we have our headquarters in West Bengal. So most of the cases we received here in Kolkata. Then second is Maharashtra, Andaman Nicobar, New Delhi, and Tamil Nadu. So this is the schedule wise animals we have identified. So total number of 565 cases, and among that 235 cases belongs to the schedule 1 species, and the rest has like less number of this schedules have less number of individual cases. So among these 565 cases, we have solved most of the cases through morphological characteristics, but 30% cases which are like were high challenging cases, we solved it through DNA molecular analysis. So yeah, total of 169 cases we solved through DNA techniques. And these are the 34 different species we have identified since 2017. And this of these 169 cases, most number of species identified are elephants, the wild boar, Indian pangolin, Chinese pangolin, and leopards. So we can kind of understand like in a particular region in India, these species are like kind of most hunted species are mostly wildlife trade. So now I'd like to discuss few of the case studies from ZSI during my time in the wildlife forensic lab. So this one is the first case, the baby chimps seized under animal trafficking. So 2018 একটা খুব ইন্টারেস্টিং কেস আছে কলকাতা পুলিশের তরফ থেকে. So they have captured few foreigners who were transporting 3 baby chimpanzees. So naturally, it was a crime. It was an illegal transport, and these baby chimps were transported to New Alipur Zoo for protection. And the zoo director, he kind of sought our help to understand like if these 3 chimpanzees are related, genetically related or not. So are they siblings or not, and if they are from same geographic region or not. So based on our genetic expertise, আমরা genetic relatedness test করে দেখি, and we found that all the baby chimps found were to be genetically unrelated. So it is possible that these baby chimpanzees are supplied from different parts of Africa, and then a single person was carrying these different chimpanzees from different parts of Africa to supply their customers in different parts. So this is one interesting case in which we used genetic technique to solve a wildlife crime here. Then the second case was very interesting, and it's kind of funny also. It's feasting on Asian palm civet and posing on social media. So there was a person from Bengal only. So he hunted some palm civet, and he posted it on Facebook, and he was showing like he has killed the animal, and Facebook online live সে দেখাচ্ছিল যে কিভাবে অ্যানিমালটাকে সে কেটে মাংস রান্না করে খাচ্ছে, and obviously then someone complained about him and police arrested this person, and then forest department they sent us this meat samples seized from this particular person, and you can see like it is very challenging to find out like which species it was, karon based on that that person will be convicted. So we do the genetic analysis to understand which species it was, and based on the genetic analysis we have found that this meat sample belongs to the Asian palm civet. So yeah, this one is a schedule 1 species, so that person got convicted in the court. Yeah, the third one is the illegal trade in pangolin. So for pangolin, we received so many samples of pangolin in different times, and it was of like different weight, starting from 1 kg to 6 kg or to 10 kg, and recently we got a seizure of like 20 kg also. So this was an interesting fact. Current pangolin, one single pangolin, either it's Chinese or Indian pangolin, doesn't matter. At a single pangolin ke mere, you can get about 900 grams of scales. So you can imagine that at a 10 kg pangolin scale consignment, it has about like 10 or 12 individuals killed in this particular consignment. Nahle eto boro consignment is not possible. So our first duty is to find out what is this species. Is it like Indian pangolin or Chinese pangolin? Then how many number of individuals has been killed, and where are these individuals has been killed, from which parts of India, or is it like outside the India or not? So for that, genetic analysis is perfect. So based on genetic analysis, in this particular seizure, we have identified this particular seizure belongs to Manis pentadactyla, that is of Chinese pangolin. So Chinese pangolin, we know it's basically from the northeast part of India. Only there they have this population of Chinese pangolin, and this seizure also came from like New Jalpaiguri. So there is a high chance that these individuals are being killed in the northeast part of India. So yeah, it is also possible that these individuals are from outside of India, but for that we need to have a different geographic samples of the species from different parts of the world, which we don't have. But for this, we have concluded that this was like of Chinese pangolin and from northeast part of India. So this is kind of like the genetic component of different pangolin species from different parts of the world. So you can see the Chinese pangolin, the Javan pangolin, Philippine pangolin, Indian pangolin, and all of them have like different cluster. So based on this cluster assignment, we can find out like where the pangolin species are geographically originated, the hunted species or hunted individuals, সেগুলো actually কোথা থেকে belong করে. Based on this geographic cluster, we can understand that. So then we tried to develop some new methodology so that it's because this is pangolin scales easily identifiable that it belongs to pangolin scale, but we are trying to find out some new methods so that people or the forest department, they can identify these scales in the crime spot altogether. So তার জন্য আমরা কিছু morphological trait নিয়ে থাকি, select করি. So we first separate the scales based on some color and size. তো আমরা ইতিমধ্যে জানতাম যে Indian pangolin scales are bigger in size and Chinese pangolin scales are smaller in size. So first of all, we did genetic analysis to find out which one are the Indian pangolin scales and which one are the Chinese pangolin scales. Then based on this genetic data, we have separated these two types of pangolin scales. And মোটামুটি প্যাঙ্গোলিনের বডিতে 3 রকমের স্কেলস পাওয়া যায়: the largest scales, then the caudal scales which are like shaped like this, and then the smaller scale or the smallest scale which are towards the tail length. So এই তিন ধরনের ক্যাটাগরিতে আমরা স্কেলস গুলোকে ডিভাইড করি, and then we do some morphometric measurements. তো আমরা length and width মাপি from every scale of the Indian and Chinese pangolin, and we found that there are some differences in their measurements, and it can be seen properly in the PCA analysis, clustering analysis. So then we have found the Chinese pangolin scales are smaller in size, the large scales of the Chinese pangolin, and the Indian pangolin scales are larger in size. So based on the size morphometry, these two species can also be differentiated in the field. Caudal scales এর ক্ষেত্রে আমরা similar pattern observe করি, যে যেখানে Chinese pangolin caudal scales are smaller in size and the caudal scales of Indian pangolins are larger in size. So our next question is how many pangolins are killed for a single consignment. So that we also used this genetic study called individual identification. So that is an ongoing process. We are trying to build up some kind of molecular chip jar basis আমরা we can easily identify the number of pangolins being killed for one consignment. So this is the last interesting case from cross-boundary identification of ivory trade in Nepal and Bhutan. So 2017 সালে we received some consignment of elephant ivory from New Jalpaiguri, and then at the similar time, so আমরা মোটামুটি পাঁচটা ছটা ivory পেয়েছিলাম. These are the length and girth size and the weight of the ivories. And on these ivories, we see that there are like blood stains. So we collected these blood stains for genetic analysis. Like in a cotton swab, we collected this blood stain and we processed it for genetic analysis. So at the same time, we received the news that an elephant has been killed in Nepal. So it was a very sheer coincidence that same time আমরা নিউ জলপাইগুড়ি থেকে এলিফ্যান্ট আইভরি রিসিভ করি ইলিগাল ওয়াইল্ড লাইফ প্রোডাক্ট, and at the same তার কিছুদিন আগেই নেপালে একটা elephant was being killed. So the natural question was, is it the same elephant or not? So this was a very interesting case where we have transboundary collaboration between the two governments of Nepal and India, and we tried to find out if this was the same elephant which has been killed in Nepal or not. So we received formal letters from the Nepal government to pursue this case, and they have sent us the elephant sample that was being killed in Nepal. So তারা আমাদেরকে blood sample and tissue sample পাঠায় সেই dead elephant. And based on that, we have our blood sample collected from the ivory that we received from New Jalpaiguri, and this is the profile of the body remains of the elephant that was killed in Nepal. So this is the genetic profile of these two samples. So if you see, like in different markers, the genetic profiling are not same. So in these three markers, you can see clearly that the genetic profiles are not matching for these two individuals or for these two samples. And based on this analysis, we have concluded that both seizures received from Siliguri and Nepal are of a different origin. So it was not the same individual which was killed in Nepal and the ivory received in Siliguri. These are two different individuals. And this is a case of identity of ascitic black bear gallbladder. So we all know that black bear gallbladder has a high demand in the illegal wildlife market because of their medicinal value or something like that. So West Bengal Baikunthpur divisioner forest official, he has seized 3 gallbladder, and he suspected it to be of black bear's gallbladder. So he sent us these 3 gallbladders for analyze and identify if this is really belongs to black bear or not. So we did the analysis, and the findings were surprising because we have identified that 2 gallbladders belong to sambar deer and 1 belongs to domestic cattle. So this is a curious case also, because sometimes what happens when a product has a high demand in the wildlife market, so there is a less supply of that product. So people try to make false product so they can sell it in the name of something. So black bear এর ক্ষেত্রে এটা সাধারণত হয়ে থাকে, যেহেতু একজন সাধারণ মানুষের পক্ষে বা একজন সাধারণ বায়ারের পক্ষে এটা জানা সম্ভব নয় যে if the gallbladder really belongs to black bear or not. So the poachers, what they do, they hunt deers or cattle, so তাদের gallbladder extract করে তারা black bear এর gallbladder বলে বেচার চেষ্টা করে. So but the main point is also here that যে যদি the DFO have suspected these are as black bear's gallbladder, and যদি সেই হিসাবে যে person who was accused তার যদি বিচার হতো, he could have been like convicted for a longer period of time. But as it is identified as gallbladder of sambar deer and domestic cattle, so there will be like just only fines or something. So this is how wildlife forensic can help in law enforcement or in the court of law to convict a person. So this is also an important case of ambergris sperm whale. So ambergris is a very interesting item. It's it has a very high demand. So ambergris আর কিছুই না, it's actually well vomit বা তিমি মাছের বমি, তাও একটা পার্টিকুলার তিমি মাছের বমিকেই স্পার্ম ওয়েলের বমিকেই ambergris হিসেবে কনসিডার করা হয়, and it has a huge demand in the market because of its aromatic components, because it is used in perfume industry and aroma industry. But this is not easy to find in nature. তো সেক্ষেত্রেও কি হয় যে প্রচুর ইলিগাল মার্কেটে এইটার ফেক মেটেরিয়াল তৈরি করে বিক্রি করার চেষ্টা করা হয়। So similarly, we have a case from Maharashtra, Thane Police, and they have sent us ambergris cost about like 20 crores. So they wanted to understand that if the ambergris is of really real product or a fake product. So it is easy to identify a ambergris product because it has a component called ambrain. So ambrain has a kind of fragrance. So jodi amra shei ambrain ke, if we try to melt it and burn it, then it will produce a different kind of aroma or scents. So based on that particular aroma or scent, it is possible to identify if it is actual real ambergris or not. So the samples we have, so it's kind of like a plasticky. So যখন আমরা drill করে sample নেওয়ার চেষ্টা করি, it was nothing like of ambergris, but it was kind of like plasticky, and যখন আমরা ওটাকে burn করার চেষ্টা করি, it gives us a plastic smell. So yeah, we have decided that this burns like black plastic, and it was not a real product, it was a fake ambergris product. The real product will have a structure something like that, molecular structure based on GCMS, but this sample doesn't have similar kind of structures. So this was the last case, this identification of wildlife trade through foreign post office. So in 2 parcels were seized by customs from Kolkata International Airport, and these both parcels were under shipment to USA. So এই দুটো parcel যখন Calcutta airport seize করা হয়, they found that there are like burned meat samples inside these parcels. So they sent it us for identification of the samples, and we did the molecular genetic analysis to find out, and it was like Oriental house rat. So the meat samples belong to rats, which was like quite surprising. But this is how wildlife forensic helps. So to summarize everything, so what I have discussed from the starting, so the basic concept of conservation genetics or wildlife conservation depends on the principles of Hardy-Weinberg. Hardy-Weinberg-এ আমরা চেষ্টা করি এটা জানার যে how the population or the species diversity is deviating from the Hardy-Weinberg equilibrium, and based on that we try to strategize our plan of action for the conservation of that particular species. And the second aspect we can use, or the second approach we can use this population genetics is in the field of wildlife forensic, and this is a very important field of study right now, because wildlife crime cases are rising in India and all over in the world, and most of the species are going to extinct if this scenario keeps going on. So wildlife forensic is a good field that can be pursued by the students, and wildlife genetics helps in this field also. And apart from these 2 approaches, there are like few more approaches, like in captive breeding program, সেখানেও wildlife genetics প্রচুরভাবে use করা হয়ে থাকে. We know that every zoo maintain their stud books, but apart from these stud books, they now follow this concept of like DNA matchmaking, which is a very new concept. So যেখানে they try to find out genetically suitable individuals for mating, which have like totally different kind of genetic variation, so that the next generation of individuals can be like more viable and genetically more stronger than the previous generation. So DNA matchmaking or this in captive breeding program is a new field of study. So সেটাও অনেকে pursue করতে পারে. And the other one is the spread of zoonotic disease. So as we know that after COVID-19, zoonotic disease is kind of a hot topic, and wildlife genetics or this concept of genetic or molecular approaches can be used in disease identification in wildlife. So particularly DNA sequencing method through the we can identify different kind of diseases that can happen from wildlife to the humans. So like right now India te Nipah virus na অনেক বেশি কাজ হচ্ছে, COVID-19 এর উপর কাজ হচ্ছে. So yes, to summarize all of this, these were the main approaches that can be followed through the concept of wildlife conservation genetics. So before ending my slides, I just wanted to discuss something for the students. So students who are in the field of conservation biology and who want to pursue this as a career option. So there are like so many options available for them. India te jodi ko PhD korte chahe, there are option for doing PhD with a different kind of fellowship. So India আমি নিজে net fellow ছিলাম. So if you want to pursue PhD in India, so net qualifying net is the best option you have. Then have gate exams. Gate exam through দিয়ে বিভিন্ন ইনস্টিটিউটে PhD করা যায় and you can have your own fellowship. Inspire fellowship আছে, Inspire fellowship through করা যেতে পারে, DBT fellowship, and then ICMR. So there are like different fellowship opportunities available in India to pursue your PhD. And there are also like different individual grant options, like you can write for your own funding, and then you can apply to some professor or some scientist for your PhD. Like I have wrote for Mohammed bin Zayed international grant, and it gives you like up to 15 lakhs of grant. So 15 lakh is a lot to complete your fieldwork and everything. And then there are like different grants like Habitat Trust, WWF, and RAFOT is an international grant you can apply. So these are the important grants you can apply for your PhD if you want to pursue this field. And for international programs, you have like altogether different fellowships. So one of the prestigious one is Marie Curie Fellowship in Europe. It's very competitive, but if you want to pursue your PhD outside, so you can apply for this kind of fellowship. Then there is like Fulbright. Fulbright is available for postdoc and PhD also. It's in USA. Newton International Fellowship in UK, and there are like in this you can take a screenshot or something and you can go and look into these different fellowship programs. I am particularly under the Swiss Government Excellence Scholarship Program. It is also a very good scholarship, and they provide scholarship for PhD also. So you can also apply for that. There is also one in Germany that is called Humboldt. Humboldt is also a good fellowship program. Then every country has their own scientific community, scientific federation. You can apply for direct grant to their federation. Like in Switzerland, we have SNSF, that is Swiss National Science Federation. So they always fund different kind of PhD programs or postdoctorate program. So if any of the students are interested, they can always apply. And you guys are lucky that you have someone like Tapujit sir and your professors who are more knowledgeable in this field. And ZSI and WII are like some good institutes to start your career in this conservation biology field, and then you can progress for like after gathering some experience in these institutes, you can go for PhD after 2 or 3 years, and in that way you will have more experience when you will start your PhD. So yeah, that was all I wanted to discuss today, and I would like to acknowledge all my lab mates and my colleagues who have done all this wonderful work. It was a great experience and a great journey with them all. And here you can see my supervisor, Dr. Mukesh Thakur. Here he is, and this is my co-supervisor, Dr. Lalit Kumar Sharma. And yeah, it was a great team and a great learning with this whole team. And I am really lucky to get this opportunity. And thank you everyone for listening to me. And if you have any question, please go ahead.
Thank you so much, sir, for this vivid and lucid presentation. I'm sure all the participants are truly privileged to have this kind of a presentation at this stage of their academics. So, and also to have the idea of the kind of fellowships that are available not only in India but globally. So thank you once again for giving your valuable time and sharing your knowledge with us. Now, I would request the participants, if you have any queries or any questions, you can directly ask our speaker. Or he has already shared his email ID with us. So if
Yes.
anybody want to contact, I think he'll be uh, very friendly enough to answer to all the queries that we have in our minds. So thank you once again for sparing your valuable time and doing this for us. Thank you so much. So
Thank you.
if nobody has any question, I would request our honorable speaker, if he could kindly share his presentation with us, if
Yeah,
that
sure.
would be helpful for the participants also. So
I have
and
a question. Can I ask?
Yes, yes, sure.
Okay. Thank you so much, Dr. Supriya, for this wonderful lecture. It was really very understanding and also very informative. I had actually one query. Like you have mentioned about the fellowship, international fellowship that student can apply for PhD and postdoc. So, what are the process by which we can apply, and what are the criteria that this fellowship required in order to get it? So, if you can elaborate, it will be helpful for us and also for the students.
Okay, so if we talk about the national level fellowships, so like CSR, NET, or GATE exams, we all know that they published the advertisement at a different time, and they have this written examination. So GATE বা NET এর জন্য preparation is very important, and I know it's difficult, but you get the opportunity to sit for the written exams, and if you clear the written exams based on your rank, you can be selected and given fellowship accordingly. So yes, and there are like national level fellowships provided by different institutes also, like NCBS such a NCBS এ যদি কেউ PhD করতে চায়, they have to sit for their own exams, but তার জন্য you have to have GATE or NET qualification. সো নেট এবং গেট কোয়ালিফাই করাটা খুব ইম্পর্টেন্ট, এবং তার জন্য যখন অ্যাডভার্টাইজমেন্ট বেরোবে ইউ হ্যাভ টু অ্যাপ্লাই অ্যান্ড গিভ দ্য রিটার্ন এক্সামস টু কোয়ালিফাই, অ্যান্ড বেসড অন দিস নেট অর গেট স্কোর ইউ ক্যান অ্যাপ্লাই টু ডিফারেন্ট ইনস্টিটিউটস লাইক এনসিপিএস অর আইজার্স অর ডিফারেন্ট ইউনিভার্সিটিস ফর ইউর পিএইচডি। সো এটা গেল হচ্ছে ন্যাশনাল লেভেল। ন্যাশনাল লেভেল ছাড়া যদি আমি ইন্টারন্যাশনাল পিএইচডির কথা বলি, তো ইন্টারন্যাশনাল পিএইচডি it depends on your communication with your supervisor outside the country. So first of all, you have to find a supervisor outside the country. So that is your job. So বিভিন্ন যে সমস্ত স্কলারশিপগুলোর কথা আমি বললাম, দে অ্যাডভার্টাইজ এট আ পার্টিকুলার টাইম অফ দ্য ইয়ার। সো হামবোল্টে যেমন সারা বছরই ওপেন থাকে, সুইস গভর্নমেন্ট এক্সিলেন্স স্কলারশিপ যেটা এসকেএস আমি যেটাতে রয়েছি, দ্যাট ওপেন্স ইন June, July, and it opens till like October. So you have like 3 or 4 months of window after the advertisement of these fellowship programs, and during this time you have to contact you have to first find an institute where you want to do your PhD or want to do your work, research work, and then you have to find a particular supervisor whose interest matching your interest, and then you have to contact them, discuss your ideas with them, and if they agree to supervise you, then both of you can write a proposal together and submit it for the funding program. So it is a basic process. So it's not easy if you are a beginner in this field. So that is why I have mentioned that it's better that if you want to do your PhD outside India, so it's better that you first gather some experience in the field of wildlife biology or whatever field you want to pursue. So তার জন্য যদি WI তে কিছু বছর কাজ করা যায় কোনো প্রজেক্টে বা CSI তে কাজ করা যায়, so that you can learn about the processes, so all the molecular aspects or the ecological aspects that I have mentioned. So তাহলে কি হবে, যদি নিজের এক্সপিরিয়েন্স থাকে, then it become more easy for you to approach a foreign supervisor, because they mostly want to see what you know and what you can give to them, what are your experiences. So based on that, your experience will help them to find a supervisor, and that is the most important part. একবার সুপারভাইজার যদি চাও, কেউ যদি রাজি হয়ে যায় to supervise you, to guide you for PhD, then it's just easy, and you just have to write the proposal, submit it, and in most of the cases few competitive grants but dile, there are most of the cases you will be selected for PhD, and you will get your own fellowship here. So yeah, that is the process, I guess.
Thank you so much. Another query I had, actually, one
Yeah.
information that I wish to have from you, because you have worked in wildlife forensic field and you have direct experience handling these situations. How much time generally? What are the duration that generally takes to solve these wildlife crimes, and how the crime branch or the government or the people involved in it are willing to solve it within the time? How much seriousness do they possess, or is there something lacking that lead to more crime or something like that? So if you can share your experience, will be very benefited.
We'll see. During my work in this field, there are like so many experiences I have, and this is very difficult. Wildlife crime and accusing someone and then bring them to in the court of justice and make them convicted is very difficult. As I have mentioned, the challenges, like sometimes we received like degraded samples, কখনো হয়তো মিট স্যাম্পল পাচ্ছি, কুকড মিট পাচ্ছি, বা জাস্ট ব্লাড স্যাম্পলস পাচ্ছি। তো অনেক সময় সেক্ষেত্রে ডিএনএ আইসোলেট করা পসিবল হয় না। যদি ইফ ইট ইজ নট possible to isolate DNA, we cannot go for like molecular approaches, and it was difficult to identify the species. তো সেক্ষেত্রে অনেক সময় অ্যাকিউজডরা ছাড়া পেয়ে যায়। Then there are cases, like it was from my experience. I was working in Nicobar Islands. তো নিকোবর আইল্যান্ডে যেটা হতো যে people have their coconut plantations there. So coconut is the main cash crop in that region, and there is this macaque species called the long-tailed macaque or Nicobar long-tailed macaque. So they feeds on this coconut. That is their primary diet, and this is a Schedule 1 species. So what the farmers do, they have like pet dogs, and they train these dogs to chase these monkeys and even kill them. So here you are not directly killing an Schedule 1 species or a Schedule 1 animal, but you are using a pet dog or a pet animal to kill those species. So how you will convict this person? I mean, the dog he has trained and he has make it run behind the monkey. It's all because of this man, and it's because of that man that the dog has killed the monkey. So how you will bring that person to the court of law? So there are like different questions also, and which is difficult to answer. And যদি আহ, বলতে চাও যে এই ব্যাপারে লোকের সদ ইচ্ছে কিরকম রয়েছে, তাহলে বলবো যে from our side we always try to generate data molecular in a molecular way or in a morphometric way. So আমি এখানে molecular approaches গুলো just discuss করেছি. There are like so many other approaches, biochemical approaches আছে, morphometric approaches আছে, যেখানে তুমি hair sample দেখে তার morphometry দেখে মাইক্রোস্কোপের তলায় medulla আর cuticular structure দেখে you can identify the species also, যেটা সাধারণত সাতু সালের ক্ষেত্রে করা হয়ে থাকে, বা অনেক অ্যানিমাল স্পিসিসের ক্ষেত্রে আমরা জাস্ট হেয়ার স্যাম্পলস থেকে অ্যানিমাল আইডেন্টিফাই করতে পারি। সো in any approaches can be used, we try to identify the species so that the person which one is accused can be convicted. But there is some lacuna in this field always, and there will be always, and that is why this field is so attractive for these criminals, because it is high profitable, and the conviction rate is very low because of lack of evidences. So yes, these are the challenges we face.
Thank you so much. You have mentioned the challenges. In
Mhm.
your opinion, what can be the solutions to the problems that wildlife crime cases faces, and how it can be solved in efficient way?
See, first of all, there should be like more strict vigilance in the protected areas, because we know that these rhinos and elephants, they are being hunted and poached from the protected areas only. So so there should be like high alert all the time, and increased vigilance in these protected areas, so that there cannot be this kind of illegal interfere of the outsiders in the protected area network. And the second one is that the government should uh, introduce some strict laws for protecting wildlife. Currently, the laws we have are like very relaxed. I mean, if you kill a tiger, you just have to give a penalty of like 20,000, 25,000, and that's it. And you will have a sentence for like 2 years imprisonment. So this like 2 years of imprisonment and a penalty of 25,000 is nothing for a person who is hunting a tiger, because this tiger skin he is selling like in millions in the black market. He is earning in millions, and he if he get caught, he has to just give a fine of 25,000 and to go for imprisonment for like 2 to 3 years, and that is nothing. So we need to have like strict laws, and killing of animals should be treated like something as serious crime as killing a human being also. So that's how we can maybe after have these stringent laws and increase vigilance, and with like proper awareness campaigns. That is also important, because I have worked in the northeast region, and in northeast region most of the communities, they are of tribal communities, and they don't have much knowledge about like wildlife conservation. It's just it is a process for them, their livelihood. So they just go to the forest, they hunt wildlife species for their livelihood only. So they don't have the concept of like wildlife conservation. So for these people, you have to make them aware about the wildlife and their importance in their nature. So yes, these three categories you can follow for combat against the wildlife crimes in India.
Thank you so much. I also have so many more queries, but I will not take so many time. Rather, I will contact you personally. Thank you. Thank you so much.
Thank you for the answers. I would request others, if you have any question, kindly unmute yourself and ask our honorable speaker. I don't think anyone have any more questions. So moving forward, I would like to request Dr. Shondip Mojumdar to kindly Assistant Professor, Department of Conservation Biology, Durgapur Government College, to kindly deliver the vote of thanks. Over to you, sir.
Thank you, Sanchari. Good afternoon to one and all who is present here with us for this one-day international webinar on molecular genetic approaches in wildlife conservation. On behalf of our college and the Department of Conservation Biology, I feel immense pleasure to take this opportunity to propose a vote of thanks. We express our sincere gratitude to our respected principal, Durgapur Government College, and patron of this event, Dr. Devnath Palik, for his constant encouragement and assistance to organize this event, and for sending his best wishes for this event. We extend our heartfelt thanks and gratitude to our wonderful speakers of the day, Dr. Supriyo Dolui, ESKS Postdoc Fellow, for accepting our invitation and to be here with us. I believe that he has enriched our participants with his excellent presentation and deliberation. We are extremely thankful to him. We are extremely thankful all the participants, students and scholars who have joined us and make this event a successful one. I also thank all the teachers and non-teaching staff of the Government College for extending their continuous support. And last but not the least, I sincerely thank all the members of our dedicated organizing team for enthusiastically organizing this event. Once again, I thank one and all wholeheartedly. Thank you.
Thank you everyone for your patience, for your patient listening. I would request all the participants to kindly take some time and uh, do not forget to fill the feedback link provided in the chat box. On filling the feedback link, you will automatically receive the certificate of this webinar. So kindly take some time out and fill the feedback form. It is already been provided in the chat box.